So buongiorno, good morning. Thank you, everyone. Thank you for being here. And thank you very much for the kind invitation to be in this Inter-Asthma meeting, World Asthma Congress. Thank you to Professor Canonica, Professor Braido, for the kind invitation. And I'm very happy with the topic I've been given for this presentation, the role of IL5 beyond eosinophils, because I think this is a developing story. And what we are going to do is to discuss the pleiotropic and multidirectional and versatile effects of IL5. That I think there is many things to learn, and the story is unfinished. So here you can see my disclosure. So we are all aware that Th2 and ILC2 cells are central players in the pathogenesis of asthma. They produce and they secrete a specific and characteristic profile of cytokines, interleukins, namely IL5, IL13, and Th2 also produce IL4. And these cytokines are responsible for eosinophilic inflammation, so-called also type 2 inflammation, either allergic or non-allergic. Classically, IL5 has been considered to act predominantly on eosinophils, not to mediate this type 2 inflammation. And it's clearly involved in the eosinophilopoiesis, eosinophil activation, maturation, and survival. But in type 2 inflammation, there are also signals, IL5 signals, from the tissue to the bone marrow that promote the maturation, the migration of eosinophils to the bloodstream, and also recruitment of eosinophils to the inflamed tissue. So the eosinophils, of course, are very important effector and inflammatory cells in asthma. And indeed, the airway levels of IL5 in induced sputum supernatant are much higher in subjects with severe eosinophilic asthma than in normal controls or healthy subjects, as shown on the left-hand side. And there is a strong correlation between the levels of IL5 in the airways, in induced sputum, and sputum eosinophilia. So there is no doubt of the strong connection, this strong link between IL5 and eosinophilic inflammation. I think this is one of the main messages, and this is part in our routine practice. We all look the eosinophilic component and inflammation in asthma. But we need also to know and recognize that there is not only an accumulation of eosinophils, but that these eosinophils are activated. A non-specific subpopulation of eosinophils have been described by Alessandra Bultaggio and Andrea Matucci from Florence, Firenze. And they have found that IL5 downregulates the CD62L eosinophils, and this accounts for L-selectin. So there is an increased number of CD62L low eosinophils in the bloodstream and in the airways of patients with severe asthma. It's important to pay attention to this subpopulation of eosinophils because this population can be modified by the inhibition of IL5 with anti-IL5 therapies. Also, there is a gradient and a further increase in the number of the CD62L low eosinophils in the nasal polyp tissue of patients with comorbid asthma and chronic rhinosinusitis with nasal polyps. So there is not only an increase of this population in the bloodstream and in the airways, but also a further increase in the upper airways, in the nasal polyps. So this population is functionally active and is clinically relevant. And which is the clinical implication of the activation of these eosinophils? Because we need to know from the clinical standpoint, which is the relevance of the elevated number of eosinophils either in blood or in the tissue. And this has been described by David Price and several authors that there is a strong positive correlation between the total blood eosinophil count and asthma exacerbations. Actually, it's one of the main risk factors to develop asthma exacerbations is the count of eosinophils or blood eosinophil count, and also the eosinophil count in sputum, for instance. So we are all aware of this important association. But also there is an inverse correlation between the blood CD62L low population of eosinophils and asthma control, as measured by ACT, asthma control test. The higher the number of CD62L low eosinophils, the poorer or the worse the asthma control. And the opposite, the fewer the CD62L low subpopulation of eosinophils, better asthma control. And here in this slide, also by Alexandra Bultayo and her team, it has been demonstrated that the inhibition of IL5 reduces the total number of eosinophils in blood, in the airways, but also the proportion of the CD62L low eosinophils in severe asthma. And this is linked to the clinical benefit, as I mentioned before, because the fewer the CD62L low eosinophils, the better asthma control. So something to pay attention, these specific subpopulations of eosinophils that are activated and have a clinical impact. But now there is evidence of the presence of the IL5 receptor in different cell types. And it has been shown that IL5 receptor is expressed by diverse, multiple cell types, potentially extending the biological and clinical relevance of IL5. And here we can see the cell types expressing the IL5 receptor alpha subunit as measured by single-cell RNA sequencing. And on the left, we can see the cells harboring this receptor, colored by cell type. We see that there are several structural, immunologic, and inflammatory cells expressing this IL5 receptor alpha. And the highest expression of the IL5 receptor alpha subunit was found in the plasma cells, also in the ciliated, you can see here in plasma cells, and also ciliated epithelial cells, but also in the mast cells. So we have evidence, at least, of the presence of this IL5 receptor alpha in a wide cell population. And for instance, if we look to some of these cells and tissues, functional IL5 receptors have been found in the epithelial cells. And the activation of this receptor by binding of IL5 influence genes of relevance for barrier integrity when stimulated with this IL5. We can see this volcano plot showing in this air-liquid interface culture of human bronchial epithelial cells incubated 24 hours with recombinant human IL5, that some genes are upregulated and other genes are downregulated. And the genes that are downregulated are mainly involved in the maintenance of the epithelial barrier, for instance, E-cadherin or caveolin 1 and 2. And these genes are also involved in the regulation of the immune response. So, according to these studies and these experiments, the IL5 receptor is not only present in the epithelial cells, can be activated and can translate signals to the genes that are downregulated or upregulated. But not only in the epithelial cells, but also in the fibroblasts, there is an increased expression of the IL5 receptor alpha subunit. And here we can see how in the patients with asthma, the IL5 receptor alpha expression is much higher than in patients that in normal controls or healthy subjects. And this is there is a several-fold difference. There's a huge difference in the presence of this IL5 receptor in the fibroblasts of patients with asthma. And in the Western blot assay, from a cell lysate of fibroblasts from patients with asthma, we can see this increased expression of the IL5 receptor as compared to normal subjects, using beta-actin as a loading control. But the IL5 receptor is, of course, expressed in fibroblasts, and this is overexpressed in subjects with asthma. And this induces the proliferation of fibroblasts. When this receptor is stimulated by binding the IL5, we can see, as in subjects, patients with asthma, there is a proliferation of the fibroblasts that is IL5 induced. And this, of course, is much higher and significantly different from normal controls on healthy subjects. But also, some specific phosphorylation pathways that are relevant in the activation of fibroblasts are activated by IL5 in subjects with asthma. We can see here, such as these two phosphorylation pathways are hyperfunctioning, are overexpressed in patients with asthma upon stimulation with IL5 in fibroblasts. So there is, in addition to an increased expression of the IL5 receptor in fibroblasts, these are hyper-stimulated and activate phosphorylation pathways. I would like to make a comment that I think is important on the role of IL5 and eosinophilic inflammation in airway remodeling. I think this is not new because this has been shown by Barry Kay and others many years ago, that eosinophils are really important in airway remodeling. But here we have the dilemma whether it is IL5 or it is the eosinophils which are really relevant in inducing airway remodeling. Can be both. Actually, both are relevant. But what is important is the net biological effect and the clinical consequences in terms of airway remodeling. For instance, and as I presented before, IL5 causes the loosening of tight junctions and increases the susceptibility to damage. So IL5, because of the stimulation of the IL5 receptor on epithelial cells, is causing this impairment or loosening of the tight junctions. And of course, the eosinophils play an important role in the formation of mucus plugs. Also, eosinophilic inflammation indirectly stimulates remodeling through epithelial damage and activation of the epithelial mesenchymal trophic unit. And this was also published many years ago by Steve Holgate. No? The activation of the M2 is crucial in the asthma pathogenesis and airway remodeling. But also the eosinophils are an important source of transforming growth factor beta. That is one of the main driving forces causing remodeling. And most of the transforming growth factor beta comes from the eosinophils. And as I also said before, IL5 directly activates fibroblasts, so by acting on the receptors on fibroblasts, which promotes airway remodeling. So here we can see the picture of the different actions in which IL5 and eosinophils can be involved in airway remodeling. And these are not all the actions. There will be more because there is a complex interactions with other cells. But something that is new, and I think it is important, is the effect of IL5 on other cell populations. For instance, in B cells, in plasma cells, because IL5 may induce, actually it induces proliferation of B cells. And here we can see on the left the heat map of expressed transcript in plasma cells from two subjects with aspirin-exacerbated respiratory disease treated with IL5, incubated with IL5. Again, some genes are upregulated, other genes are downregulated. But the genes that are upregulated are involved in cell cycle and in growth-related pathways. This means that the IL5 from plasma cells or antibody-secreting cells is causing proliferation of plasma cells. Actually, it also promotes the synthesis of heavy chains, including for IgE. So we have this local production of antibodies and stimulation of the immune system. And on the volcano plot on the right side of subjects with chronic rhinosinusitis with or without nasal polyps, and we can see that in those subjects with aspirin-exacerbated respiratory disease, that is a severe form of the Sumter syndrome, no? We can see an overexpression of the IL5 receptor alpha and also of the CCND2, indicating the importance, again, of the expression of the IL5 receptor alpha in patients with comorbid nasal polyps, and in this case also particularly in those with associated aspirin-exacerbated respiratory disease. Another aspect that I think is very important is the interaction between mast cells and eosinophils. To me, this is a clue finding and of utmost importance. It's really relevant because mast cells and eosinophils work together. This has been called the allergic effector unit. They are not only, do not only have physical interactions because they are in the vicinity, they are communicate through soluble mediators. For instance, mast cells produce and release a variety, an array of different enzymes, chemokines, interleukins, autocoids, eicosanoids. Some of them are going to stimulate the eosinophils, of course. For instance, leukotrienes, prostaglandins, and the eosinophils are going to release the granule proteins, basic proteins. And it has been shown that the major basic protein and the eosinophil cationic protein are able to stimulate the mast cells and to increase the produce of prostaglandin D2. And this can be inhibited and blocked acting on a specific receptor called the MRGPRX2 receptor. And so the crosstalk and the interactions between mast cells and eosinophils are of greatest importance, particularly when mast cells are located within the smooth muscle bundles, because that's one of the hallmarks of asthma. Not only the number of mast cells, but also the location and the interaction and the dialogue with the eosinophils. So I will say that briefly, but in summary, IL5 orchestrates multidirectional effects on structural and immune cells. The best known effect is on the differentiation, proliferation, survival, and migration of eosinophils. And this, of course, contributes to increased eosinophil activation in blood and tissue that has clinical implications and that we are using on routine clinical practice. But there are additional effects. IL5 downregulates genes related to tight junctions and barrier function in ciliated epithelial cells and modifies epithelial activation. And it also enhances the proliferation and activation of fibroblasts, mediates smooth muscle cell activity. Actually, the IL5 receptor alpha has also been found in smooth muscle cells in the airways, mediates ILC2 cell activity, mediates T-regulatory cell production, upregulates genes involved in plasma cell proliferation, and also it produces an increased expression of the IL5 receptor alpha subunit on an activation of neutrophils, but we don't know the clinical relevance of this activation in the neutrophils. And it enables the mast cell and eosinophil crosstalk. And this is just part of the picture. There are other actions. And this is my wrap-up slide. I think it's important to consider the evolving evidence suggesting that IL5 has an impact beyond eosinophils, leading to epithelial barrier dysfunction, airways remodeling, and disease progression. This is due to the multidirectional effects of IL5 that orchestrates effects on immune and structural cells. Among the structural cells, the epithelial cells, the fibroblasts, the smooth muscles, inflammatory cells, the eosinophils, immune cells, T-regulatory cells, ILC2 cells, but also plasma cells, neutrophils, mast cells. So there is an array of cells that are being activated by IL5 beyond the eosinophils. This translates into several clinical manifestations and disorders. For instance, nasal polyp formation, immune imbalance. We need to consider that these diseases are due, in the end, to the immune imbalance that is being caused by different triggers or agents. Epithelial barrier dysfunction, that is very relevant in asthma. Mucus plugging, also relevant in airway remodeling, and airway remodeling itself with all its components, particularly the activation and proliferation of fibroblasts. Of course, this leads to disease progression, both in the upper and lower airways. So I would like that my take-home message is this one. There is evolving evidence suggesting that IL5 orchestrates multidirectional effects on immune and structural cells, leading to epithelial barrier dysfunctions, airways remodeling, and disease progression. Thank you very much for your attention.